All right, so let's talk about the other organelles now. Organelles in general. We have the nucleus. Some cells may have more than one nucleus, and that's how they make it plural, nuclei. All right. This controls the activities of the cell. This also is the thing that's going to hold the DNA, the blueprints of you, the Constitution to the United States. It needs to be very well protected. So there's a double membrane to it. Okay? We also have something called a nucleolus. And some cells have more than one nucleolus, and we say there's nucleoli. Just word alert, just be careful with spelling. Big issue here. Okay? Plural, singular, which one are you talking about? Big mistake. Well, anyway, the nucleolus is found inside the nucleus, and this is what makes the ribosomes. Okay? It has its own, what we call ribonucleic acid, and it makes the RNA also. All right, so we'll talk about that. Part of it makes the ribosomes. We'll get into it, but it makes the RNA. We also have the ribosomes, those tiny balls that are scattered throughout the whole village. These are the ones that are actually going to make proteins. All right, these are the little shops that are doing that. This is something, as I mentioned before, that does not have a membrane around it. In fact, it's made of two parts. And think of it like a big clam. Okay? So they're scattered around there. We also have the endoplasmic reticulum, and that's the roadways through the village. All right? That's going to transport materials from one place to another. Golgi apparatus, also known as the Golgi complex. That packages and stores the protein so that it can be used later on or that it can be used right away. So it kind of packages it. Okay? It's like you make all the, if you want to make all my PowerPoint presentations and print them on paper, fine. You just made the protein, you just printed them all. But you want to put spotted round holes over there and package it so it's easier to carry, right? So that's what Golgi apparatus does. It kind of packages the protein. The protein's already made, it just puts it into a form that's easier to manage. Lysosomes, these are your police of the village, okay? They contain these digestive enzymes that will, the lysosome will fuse with an intruder that comes in, a virus, a bacteria, whichever, and will release its digestive enzymes and destroy that bacteria. Okay? Also, when the cell needs to die, it will just release its, all its digestive enzymes, and all lysosomes do that, and it's like self-destruction. It just will eat away the cell itself. Mitochondria, they will contain the ATP. This is the one that's going to be the powerhouse of the village. It's the one that's going to make all the energy. We got a whole chapter or two just on the mitochondria. That's next week. All right, we'll deal with that next week. We also have the chloroplasts. Again, this is only found in plants and algae. Okay, I think some bacteria also have it too. All right, but not in us. All right, this is the site of photosynthesis, and it has a green pigment called chlorophyll. And we got a whole chapter in just photosynthesis. Okay, and we have vacuoles. These vacuoles are only found in plants, fungi, and some protists. And I kind of introduced you to a vacuole, contractile vacuole that a paramecium has, where it takes in a lot of water, but the water doesn't go inside the cell. Well, it does, but immediately goes inside this vacuole. The vacuole gets bigger and bigger and bigger. When it gets to a point, it just shoots out all the water out of it. So it pumps out the water. Okay? So let's spend a little more time talking about each one of these organelles, and then we'll be done. Okay? So the nucleus. The nucleus itself is going to be carrying the Constitution of the United States. It's the thing that's our DNA. And the DNA will never, ever, ever leave that nucleus. The Constitution will never, ever leave the Washington, D.C., or wherever it is. Is it there? Which is a part of Washington, D.C. All right, so you can't take it out, right? So that's where that is. all right? We could make copies of it, right? People have copies of the Constitution in their back pocket. You can make copies of it, but you're not going to get the original, all right? That stays in. Since it's so vital, we're going to have two membranes around it. We call it a nuclear envelope, all right? And they're contained, we have these little pores. The pores are going to allow certain things to come in. You need a backstage pass to get into the nucleus. Okay? And we also have chromatin, which is really the DNA, the alignment of the DNA. Chromatin and chromosomes are the same thing. When you hear chromatin, that's DNA. You hear chromosomes, that's DNA. What makes them different? One's coiled, one's uncoiled. What does that mean? Okay? When you can see, when it's coiled, you can see it. When it's, and you can see it like closer with a microscope. When it's uncoiled, you can't see it. All right, big deal. Why does it do that? All right. Think of it like this, and we'll get into this too later on. Let's say you have a 1,000-page book or encyclopedia. Do you guys know what an encyclopedia is? I don't know. Oh, okay. Now it's just called Google. All right. So you have this 1,000-page encyclopedia. Now, it's all condensed into one bound thing. It's easier to have it in a bound thing, and you can separate it. If there's, like, say, 46 encyclopedias, like volumes of encyclopedias, 46 chromosomes, 46 volumes, okay? They're easier to carry when they're packaged together like that, when they're condensed. And you could separate it. Here, here's 23 for you and 23 for you, whatever you want to do. They're easier to split that way, right? And when it's in that form, we call it chromosomes. And we can see that when we do obvious things like mitosis. And that's when we're splitting things up. But in most times, it is not coiled. It is not packaged. It's loosened. It's taken out all those pages, every single one of those pages, and laying them down across the whole floor in this room. Very thin. But what's nice about this is that if someone asks me, I need to do a paper on, I don't know, Roman architecture. I could scan through this whole thing, because I can see the whole thing and say, oh, there it is. It's much easier to pull out information when it's very thin. You can't pull out information when it's condensed. Does that make sense? All right? So when it's condensed, it's going to be easily split up. Those are chromosomes. Most cases, the DNA is in chromatin form. Very thin, uncoiled, so that when we need to make a protein or a recipe, we'll get into those later, it's easier to manage. You can see where everything is, and you can pick exactly what you want to make a copy of. Does that make sense? The difference between chromatin and chromosomes. They're the same friggin' thing. It's just one is coiled, meaning it's condensed, and we can separate it. Other times it's thin, and we can read everything, and we can pick out things, and it's much faster that way. We clear with that? Okay. The nucleolus is there. That's the site of ribosome and RNA synthesis. Okay? You can see it right over here. And like I said, the nucleus is what's going to dictate what the cell is going to do. Okay? Some cells don't have a nucleus. They did at one point, but certain mature cells don't. We've been talking about the red blood cell quite often. A mature red blood cell is floating throughout your whole body. They don't have a nucleus. Okay? And the reason, in a very big nutshell, is that red blood cells carry oxygen, or hemoglobin. If you put this big nucleus in there, well, that's going to keep up too much space. Why don't we just take the nucleus out and fill that space with more hemoglobin, more oxygen? It's more efficient. The problem is with red blood cells, if they don't have a nucleus, they die. And they last for about 3 to 4 months, and then they die. So our bodies always have to make new red blood cells. They won't go through mitosis, okay, which we'll get into later. Multinucleate. There's some cells that need to make a whole slew of proteins. Certain muscle cells, muscles in your arms, they have many, many nuclei there. So where they're multinucleated, okay? And also a certain cell that's in the bone, something called an osteoclast. Right? If you've heard these words before, I want to ask you, but if you heard them, I'm trying to put them in. Ribosomes. This does not have a membrane around it. It's more like a clam, 2 parts. There's a small, what we call 40S subunit, and there's a bigger 60S unit. So you have a small unit and large unit. They kind of clam together, and you got yourself a ribosome. So there's structures that don't have this membrane around them. Okay? Now, a lot of ribosomes are just free-floating in the village. Some of them, in the village I was using shops, like wood shops and metal shops. All right? They're going to be making things. Does that make sense, right? They're going to make a doghouse or whatever. So ribosomes are the places that are going to make things, specifically proteins. Now, some of them are away from the roads, right? You know some stores that are not on the road, they're freestanding someplace else. You have to walk over there, right? Just the woods or whatever. Exaggeration. Okay? So some of them could be free. Some of them are right attached on the roads, right on the endoplasmic reticulum. There's a Staples that's way off the main road. But some Staples, it's right there on the main road, right? So think of it like that. And Staples does a lot of building, right? They make copies of things. So the ribosomes, they carry proteins and RNA, rRNA. I'm stuttering. We'll talk about those another time. So the ribosomes are going to read our genetic codes by way of something called mRNA. All right? We get into building a protein, it'll make more sense. What they're going to do is they're going to assemble amino acids in a certain order, and I showed that briefly before. When you put a bunch of amino acids in a certain order, they're going to make proteins. But it has to be done a specific way, right? Alanine, if it's supposed to be alanine, comes before histine. You can't put a valine there, otherwise that's how sickle cell anemia develops. Endoplasmic reticulum, the roadways. They're interconnected, but somehow they're all connected to the nucleus. Same way as Washington, D.C., all the roads somehow, doesn't matter where you are in the United States, with the exception of Hawaii, somehow you can still get to Washington, D.C. There's 2 types of endoplasmic reticulum. There's rough endoplasmic reticulum. And this is where the ribosomes are on the road. They're on the endoplasmic reticulum. They're all beaded on there. They're going to contain many ribosomes, and these are the ones that are going to be mainly concerned with proteins for obvious reasons, the ribosomes are there, and also making phospholipids. So it's not just roadways, they also are responsible for making the phospholipids. We also have smooth ER, smooth endoplasmic reticulum. These do not contain the ribosomes, but these are responsible for making lipids. Okay? The Golgi apparatus, also known as the Golgi complex. They look like stacks of envelopes. And these are the ones that are going to package the protein that you just made. Okay? Again, my analogy was, you want to make copies of all my presentations, and it's on paper, you get it done at Staples. You made the protein. You made the copies. But you want to package it so you can take it home and deal with it later, right? You put holes on the sides and put it in a binder, whatever you got to do. That's packaging. So the protein's already made. So they can get released, or the protein can get released inside what we call a secretory vesicle, the actual bound book. You won't put it around saran wrap and stuff. It's in a vesicle. And there's 3 fates that protein will have once it leaves the Golgi apparatus. It'll either become a secretory vesicle for later use and just stay inside the cell. That's probably what you guys may have done, made copies of everything and of my PowerPoint presentations and just put it off to the side for a later use, right? I'm not getting laughs, okay, good. Some may migrate to the plasma membrane and become a part of it, right? We made proteins. We know that some proteins are a part of the membrane, so it could be embedded into the membrane. Some may just shoot out of the membrane to go someplace else. Whereas other ones can become lysosomes. There's proteins, digestive enzymes. Remember, an enzyme is a protein. And giving it to the security guard in case there's a criminal that comes in. So this is just showing you the 3 fates over here. There's the endoplasmic reticulum, the blue. Here's the Golgi apparatus in green. We can either turn it into a vesicle, it'll shoot out all the proteins or whatever it needs to do outside, exocytosis. It could make a protein and become a part of the membrane itself. Or we could just use it later. It could either turn into a lysosome where there might be a criminal coming in that we could use later in that sense, or just a vesicle that will use the protein, maybe not lysosomes, but other things. We could use it for a later use. Lysosomes. These are going to protect things from coming inside, right? So they contain these powerful digestive enzymes, bullets and guns, right? And they're going to digest anything that comes in, any intruder that comes in, whether bacteria or virus. Now, granted, if you're going to have a lot of bacteria, a lot of viruses come in there, you know, there's so much that the lysosomes can do. They'll get overwhelmed, and then the bacteria will just overwhelm that, and the virus will, and then you get sick, right? It'll also degrade any non-functional organelles, kind of deformed organelles that just didn't make it, so we're going to just destroy them. Or, like I said, if the cell has to decide to self-destruct, it's just getting too old, then we'll do the cell suicide and the lysosomes will just burst open and digestive enzymes go all over and just destroy the cell. We call it autolysis. Auto meaning to oneself, lysis, and you heard that before, to break up, to degrade. Brings up another disease, Tay-Sachs disease. Who here has heard of Tay-Sachs disease? Question, or you've heard it? You heard of it, okay. Nobody else has heard of Tay-Sachs? You've heard, okay. Yeah, a few of you have heard of it. Yeah. Tay-Sachs disease is a disease that's usually found in the Hasidic Jews. Okay, we see a lot of it there. Hasidic Jews, if you see they have the curls over here, that's a really, really strict Jewish culture. So it's a genetic disease. They're not the only ones that get it. There's other ones, but we just see a predominant in that population. So we have a lysosomal enzyme, all right, that all of us have. It's called hexoamidase. We all have that. But in Tay-Sachs disease, it's not developed. It's not there. It's deficient. This hexoamidase usually will break down glycolipids in the brain. for another reason, they just build up and build up, and we need to break them down, and then we could use the parts later on. We don't want to be building up. So in all of us who don't have Tay-Sachs, it breaks it down no problem, right? Well, in people with Tay-Sachs disease, this glycolipids start building up and building up. We can't break it down. So you're going to have a lot of glycolipids building up in there. There's a lot of nerves that are going in the brain, and that glycolipids are, since there's so much outside the nerves, it starts pressing down on the nerves. And it interferes with conduction. It's kind of like if I have a hose over here and water's going through it, no problem. But if my body is glycolipids and we have too much of it, I'm stepping on it. Am I going to be able to get the water through there? No. Well, think of electricity going through there. All right? Or the nerve impulse. You're pressing down on it. So so if this is a nerve going down here, all right, and this nerve goes to the eye, if there's pressing down on here going on, is there going to be electricity going to the eye? No. So that's going to cause blindness. You see what's going on? So there's a lot of things that affects a lot of nerves in the brain. So you have loss of coordination, dementia, just not remembering things like that. And these are just some things. But the problem is there's death by 5 years old. It's a pretty severe disease. Okay, and that's Tay-Sachs, and it's all dealing with a lysosomal enzyme not having it. Okay? Peroxisomes. They're similar to lysosomes, except there's only one thing inside of it: hydrogen peroxide. All right, I'm sorry, catalase. Catalase. Hydrogen peroxide is damaging to a cell. That's why if you get an infection, you put hydrogen peroxide on there to try and kill off any bacteria there. Bacteria doesn't like that. We don't like it either. But for some reason, if we have hydrogen peroxide that's building up, we need something that's going to break it down. Hydrogen peroxide has a formula of H2O2. And what catalase does is takes the H2O2 and turn it into oxygen and water, stuff that we can use, so that it's not going to be debilitating for our own self. Okay? Questions about this? Okay. Mitochondria, the powerhouse of a cell. Very briefly, let me talk about it. This is what makes up the ATP. There's two membranes here. The outer mitochondrial membrane, and then there's this yellow portion that kind of undulates in there. We call that cristae. And that's another membrane. That membrane is very important for what we call cellular respiration. And there's 3 parts to that, and the last part is, which is called electron transport chain, is going to be a part of that. So we'll be talking about the mitochondria probably next week. Okay? Interesting about mitochondria, because they have to make ATP, and ATP is vital, right? If your body makes one circuit of ATP and it doesn't make anything more, you'll only live for about a minute. It always needs to make more ATP, even when you're sleeping. So this is what's neat about mitochondria. It doesn't want to wait for the nucleus to say, do I need to make more ATP? It kind of gave them the waiver and say, you know what, don't worry about us. So we got the nucleus, I'm the nucleus, I got a lot of other things to be concerned about. All I want you to do is make ATP and continue doing that. So you know what, I'll give you some DNA. So mitochondria has its own DNA, has some of the original constitution in there. Because it's such a vital part of, if you don't have ATP, nothing's going to work. So they don't want to hold off. They don't want to wait upon the nucleus to tell it what to do. It has its own DNA to do this, mitochondrial DNA to do this. So it has its own RNA, it has its own DNA. Is that clear? Okay? So So they can even replicate themselves. If for some reason they're low on, they need a lot of energy, and they need to make more ATP, the mitochondria just decide on themselves, you know what, let's divide our cells and make more mitochondria. We don't need the nucleus's permission to do that since we have our own DNA. All right? So that's what happens with that. So all energy in our body is ATP, as you know, right? Adenosine triphosphate. And there's two main ingredients that you need: oxygen and glucose. You don't have, if you starve and you don't get glucose, you die. If you suffocate and you don't get oxygen, you'll die. You need those two things. Sure, you need everything else too. You need sodium and chloride and other things. Yeah, that makes the machinery run. But again, to make potato chips, the two main ingredients is potatoes and oil. You need bricks. It's not going to go in there, but it's the machinery, right? You need bricks to make the factories. You need the metal to make the machinery. You need people. So yes, you need salts, you need other things. But the two main ingredients that you need a constant supply of is glucose and oxygen. And the mitochondria will take these things and convert it to ATP. Okay? So let's talk briefly about mitochondrial DNA. All right? When a sperm enters an egg, all right, the sperm's mitochondria doesn't enter the egg. Interesting. The egg has mitochondria and has a nucleus. The sperm has mitochondria and has a nucleus. And the sperm also has a tail. But when the sperm goes inside the egg, the tail breaks off. The tail never goes inside. But also the mitochondria gets destroyed. So that the nucleus of the, which is the main thing that that egg needs, is the nucleus, goes inside the egg. So the mitochondria does not get passed to the offspring from dad. Does that make sense? Mother's does, though. So that brings about some mutations. Right? DNA has a lot of mutations. We talked about insertions and things. Well, the mitochondrial DNA mutates more than nuclear DNA. They produce certain rare diseases, some muscular diseases you really don't have to worry about right now. But think about what's happening here. That if dad has one of these muscular diseases that are due to mitochondrial DNA, can his son get it? Can his son be a carrier? His daughter? No. Unless mom has it, because mom will pass down the mitochondrial DNA. Does that make sense? All right? So for Mother's Day, you could have a card that says this. It's a beautiful saying to put it. Mom, you gave me something Dad never could. They won't understand it, but you would, right? It's one of those Far Side kind of cartoons. All right. Just about done. Chloroplasts. Okay? They're only found in plants and algae and a few bacteria. Okay? And what this is a site for photosynthesis. It captures light energy and converts it to chemical energy. That's a fascinating thing. How do you get light energy and it turns it into chemical energy? That's another talk for another time. All right? But you get this light energy and it makes sugar from carbon dioxide and water. Okay? So plants are very interesting. They make their sugars that they'll use, so they make their own food. They make their own energy. All right? Whereas we can't do that. We have to survive off of other things. We can't make our own food. Does that make sense? They can. They've done it for billions of years and it works. A chloroplast is what we call a type of plastic, which is an organelle that makes and stores pigments on food molecules or for food molecules. All right? So that's what gives the fruit color, the flour color, starch, lipids, and so forth. It's a double membrane, all right? Contains their own DNA and contains chlorophyll, which is this green pigment. So it's very similar to mitochondria. The only thing I want to say is that plants have mitochondria and they have chloroplasts. Chloroplasts make the food, the mitochondria will take the food and turn it into ATP. Okay? So, it's weird, but what happens with chlorophyll, or not chlorophyll, but with the chloroplast, it makes ATP, but all that ATP is used to make more ATP. And then that ATP is used to run the whole thing. We'll get into that later. And this chloroplast contains enzymes and other molecules to help function with photosynthesis. Okay? So this is what it looks like. This is a chloroplast. There's many chloroplasts in there. It's filled with chlorophyll. And what it looks like is that we have these little pancakes, green pancakes over here that are stacked. Each green pancake is a thylakoid. When you got the big stack of them, that's called a granule. And these stacks of pancakes are connected to each other. See that there? The only other thing we have around here is the open space that's surrounding these stacks of green pancakes. The aqueous solution around there. That's called the stroma. There's certain things that happen in the stroma. There's certain things that happen in the thylakoid area. And we'll get into that with photosynthesis later. Vacuoles. They're found in plants, fungi, and some protists. We mentioned one with the paramecium. So you could have food vacuoles. They're formed by phagocytosis. All right? We could have contractile vacuoles. Like I said, parameciums are freshwater protists that if water goes in there, the water goes into this contractile vacuole and then pump out all the water so it doesn't destroy the cell itself. Central vacuole, we have them in many plants. It's large. It's a storage area for organic compounds and some water. Okay? And that's it.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi Apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.